Okay, so I think we are ready to get started. Hi everyone, and welcome to the digital expert event hosted by Vator Securities. My name is Christopher Krolak. I'm a partner here at Vator Securities. Today we have the pleasure to host the event on behalf of NextCell Pharma that recently announced positive results in its phase 2 trial called ProTrans 2, showing a statistically significant improved preservation of insulin production in type 1 diabetes patients. For this event, we have some very distinguished guests. To start off with, we have the CEO of NextCell Pharma, Mathias Svahn, who will present the study results and the roadmap for ProTrans. We also have cell therapy specialist Dr. Weiss that will give an introduction to mesenchymal stem cells and the significance of the study results. We also have diabetes specialist Dr. Landin-Olsson, who will elaborate on the clinical relevance of ProTrans and the value of preserving patients' endogenous insulin production. At the end of the event, there will be a Q&A session moderated by Felicia Rittemar, who is a healthcare analyst here at Vator. So if you have any questions, you're more than welcome to send them in via the Q&A form that you will find at the bottom of the screen. So with this short introduction, I would like to hand over to Mathias Svahn, CEO of NextCell Pharma.
Thank you very much. I will turn on my slideshow here. Thank you all for tuning in. I'm very honored to be joined by Dr. Weiss and Dr. Landin-Olsson to present our data and have an independent evaluation or reflection of the results that we have generated. So just a brief introduction. You hopefully know NextCell Pharma well by now. This is a disclaimer. So NextCell Pharma is a cell therapy company that has the lead drug product called ProTrans. And we are currently evaluating the efficacy and safety of ProTrans in type 1 diabetes, which is the first indication. ProTrans is a mesenchymal stem cell therapy with the aim to reduce autoimmune diseases and also for preventing inflammatory states. So we have this currently ongoing in clinical trials in type 1 diabetes. And in addition to that, we have a project where we are ready to enter clinical trials and yet another indication where we're still evaluating in preclinical setting. In addition to that, we have Cella Viva, which is a stem cell bank, but we will focus entirely on ProTrans today. So the scientific rationale for using mesenchymal stem cells for autoimmune diseases and other inflammatory states is that these cells have a balancing role in the immune system. They interact with all kinds of immune cells to have the balance between inflammation and regeneration. And they interact through various pathways. In the right-hand panel is a schematic of diabetes type 1, which is an autoimmune disease where the patient's own immune cells starts to attack the insulin-producing cells in the pancreas. And there are other immunosuppressive treatments being evaluated presently, which focus more on the immune cells themselves, where we use or they use antibodies to knock out specific immune cells. Whereas we use mesenchymal stem cells since they have this balancing role and can affect multiple different types of immune cells, which makes this a wider application and the safety issues are less concerning than when you immunosuppress. The core of ProTrans is really how we assess the cells. We had developed a selection algorithm to evaluate the cells used for ProTrans through their potential to affect different pathways within the immune system. We look at the interaction with T cells. We look at the expression of prostaglandin 6, prostaglandin 2, IL-6. We look at the IDO-fold induction, and we make an overall assessment, because we don't know every time what the driving mechanism behind is, but we know that these cells are involved in the autoimmune disease. Therefore, we want our cells to be able to respond in various pathways. And we have evaluated our drug in clinical trials. The first trial was ProTrans 1. It's a dose escalation trial where we treat newly diagnosed adult patients that still have a remaining endogenous insulin production. It is important that they still have that, since our cells will not produce insulin. They are given to the patient to reduce the autoimmune response towards the insulin producing cells. So in this first this first open label dose escalation trial, we treated 9 patients, and the primary endpoint was safety. We treated 3 patients with low dose, which is 25 million cells, single injection, medium dose 100 million cells, and high dose 200 million cells. All these doses were tolerated. We have only 2 adverse events during infusion. One had headache and one started to smell like corn, but that is more adverse event from DMS of the cryopreservant than to the drug itself. So it was very well tolerated. We also looked at if there was a signal for efficacy, dose-dependent efficacy. And in the panel to the left, we did detect a statistically significant difference between low dose and medium dose, as well as low dose and high dose. And there was no significance between medium and high dose. So when looking at the percentage in insulin drop after 1 year, which is the right panel, we put the high dose and medium dose in the same bar, which is the blue. And we saw that these patients, they had a reduction in insulin production in the level of 8%, as compared to the low dose patients that had a decrease in close to somewhere 25-30%. So that was very exciting, and to some extent it was also I mean, it's a very little material, so we were very happy to find significance in such a small material, but that also you have to be cautious about these small numbers. So we went for a randomized, placebo-controlled, double-blinded trial with 10 patients receiving active treatment. We chose high dose since we didn't have any safety concerns in any of the dosings. And we compare that with 5 patients in the placebo group. The inclusion criteria are identical. The most important part here is that all patients are randomized. Neither the patient nor the doctor knows if the patient got treatment with ProTrans or placebo. And we give a single infusion and then evaluate after 1 year. The primary safety endpoint is still adverse events. Primary efficacy endpoint is the insulin production as measured as Cpeptide, area under the curve, before treatment as compared to 1 year after treatment. And the primary endpoint was achieved. We saw a statistically significant difference between the patients receiving ProTrans. In average, these patients lost 10% of their insulin production in comparison to the placebo group, who lost, in average, 47% of the insulin production. And this was then analyzed per protocol as a 2-sample t-test. So with that, I would like to give the word to Professor Mona Landin-Olsson, who is a senior consultant at the Skåne University Hospital and a professor at Lund University. Thank you for joining us.
So thank you. Yes, can you change the slide? Yes, thank you. So I'm going to talk about diabetes as such and the relevance of Cpeptide and remaining insulin production in the patient. The Cpeptide is a rest product from when you are doing insulin. You have a big molecule of proinsulin, and then you cleave off the Cpeptide and you use the insulin. So the insulin is metabolized, is taken up by the liver and the muscles, and you have a Cpeptide circulating, and then it goes out by the urine. And it's believed that Cpeptide as such has no effect. There has been some claim that it should have an effect for the development of neuropathy, but that is unsure. So Cpeptide is mainly a good way to measure the endogenous produced insulin. And you can distinguish the insulin that you are giving by injections, the exogenous insulin administration. So Cpeptide is made by the patient himself. Yes, so we can take the next slide. Type 1 diabetes, the etiology is not quite clear. It's believed that it is an autoimmune disease, that the immune system attacks the beta cells and destroy them. And on the basis of this knowledge, lots of trials has been started and are ongoing. And these are some of the trials: CD3 blockers, CD20 blockers, and TNF-alpha, and anti-thymocyte globulin. And all these trials are the main aim is to block the immune system and stop the destruction of the beta cells. And when you are evaluating these studies, it's extremely difficult to evaluate, because when you have a patient, there is a different velocity where you destroy the beta cells. So you can have a rapid destroyment, and you could have a very slowly progressing disease. And when you measure the C-peptide, you can have high levels of C-peptide, or you can have a very low. And in this study, you should have some remaining C-peptide. That means that you still produce insulin. And after 1 year, you could have a recovery of the beta cells. That is called the honeymoon. So when you have a blood glucose that is normal by insulin, the beta cells is recovered, and they can start to produce a little bit of insulin. And then you have a falling curve, and after a couple of years, you have no insulin left. But depending on when you come in this phase, it's very difficult to say that one patient do better than the other patients. So it requires quite big numbers of patients to be sure that it has any effect. So we take the next slide. So these are the results, and these results are based on quite few patients. And the blue one, that is the active cells, and placebo in the red. It seems that there are differences, but on these very few patients, I will say that the results are very unsure, but it's still results, and they are worth to do in a bigger scale, I think. Yes, next, please. And these are also based on the randomized study with 9 evaluated patients in the active group and 5 in the placebo. And then there is added the patients from the escalating study. So it will mean that there are 15 patients, and the placebo patients are 5, I think. 8 patients. Yes, the results are very unsure, but they are still, it looks promising, but you can't be sure that this should be something if you have a bigger study. Yes, next please. And this is the importance of a C-peptide. If you have a C-peptide, you have a better outcome. So if you are able to produce own insulin, you have a buffer against very high values and very low values, and you could add insulin. And we know that from studies from transplanted patients. If you transplant the patient with pancreas or beta cells, and they have a small little own production of the insulin, you have it's much easier to have a good metabolic regulation of a blood glucose, and that is of importance for the complications, the development of complications. So if you can't cure the patient, so he will get back all the insulin production, it's good enough to have remaining insulin production. That is for sure. Yes, thank you. There was perhaps some yes, reflections. So we have no treatment. We have no curing treatment. We have only symptomatic treatment for type 1 diabetes. So it's very important to take to get other ways to treat type 1 diabetes. And all the immunosuppressive treatments have serious side effects, and the results has not been so good. The number of patients is crucial because we need a lot of patients to be sure that the effect is as it shows. And I think this is you should continue to have a bigger scale up these experiments. Yes, thank you.
So thank you very much, Mona. And now I'd like to introduce Daniel Weiss, Professor Daniel Weiss from University of Vermont, in professor at many places, and also a former chief scientific officer of the International Society for Gene and Cell Therapy. So it's really a pleasure to have you here with us. Thank you, Dan.
All right, well, thank you, Mathias. And I appreciate the opportunity to present some more background information on just what mesenchymal stromal cell therapy is all about. So if you could go to the next slide, I'm going to present information that some of you in the audience may very well be familiar with, some of you may not, but it's always good to hear it again and place the context of the NextCell product in its proper place. So we're talking about mesenchymal stem or stromal cells. They're known as both, and they're originally described as stem cells. So if you look at the schematic in the upper left-hand corner, these are cells that were originally found in bone marrow, but now are well recognized to be present in other tissues, including adipose tissue, and very importantly, cord blood, Wharton's jelly, placental tissues, which is the source of the NextCell product, if you will. These cells can act as stem cells in terms of their being able to differentiate into specific tissues, most notably bone, cartilage, and fat, adipose tissue. But what we're talking about in terms of the biology and the behaviors of these stem cells has nothing to do with their stem cell capacity. We're not looking, NextCell is not looking for these cells to act as stem cells. Rather, they have immunomodulatory properties. And to understand that is a schematic in the lower right-hand side. And this shows the bone marrow and the cells that live in the bone marrow. And what you can see is that the MSC, the green cell that is living along the outside of a blood vessel. And what the MSC does, and it's the same from cells isolated from bone marrow or adipose or umbilical cord blood, is that they express on their cell surfaces receptors that will sense inflammation. And so if the cell senses a certain pattern of inflammatory stimuli, for example, those that might be present in type 1 diabetes, then they will release in return a portfolio of different anti-inflammatory mediators. And these will include things like anti-inflammatory cytokines, antibacterial peptides, extracellular vesicles, and a whole range of things that we're still trying to better understand. But the goal is that the cells have evolved to counter the inflammatory situation in any given situation. For the bone marrow, it's felt that this helps the bone marrow protect the hematopoietic stem cells that need to differentiate into red blood cells, white blood cells, and platelets. So next slide, please. In terms of the immunosuppressive properties, so Mathias had touched on this initially, but these cells will secrete substances that will both inhibit the proliferation, so the growth of different immune cells, things like T cells, things like B cells, and others listed. And they'll also inhibit the specific functions of those cells. So for example, if a T cell is overactive and is part of the pathogenesis of destroying the islet cells resulting in type 1 diabetes, the MSCs, and the goal for the MSCs is to inhibit that function, inhibit the T cells, and protect the beta cells, the islet cells, so, and maintain insulin production. Next, if you hit next, please. So immune regulation, hit next again, is the potential rationale for use in not just type 1 diabetes, but a broad range of autoimmune and inflammatory diseases. And again, we're not asking these cells to behave as stem cells. We're not asking them to differentiate, rather we're utilizing this other property that they have. And this is quite relevant for type 1 diabetes. Next slide, or the next, please. And so from a practical standpoint, how does this work? How can you imagine taking cells from one individual or cord blood, in the case for NextCell, and giving it to a person? Why wouldn't that cell be rejected by the recipient, by the host immune system? So it turns out that the MSCs do not express on their cell surfaces proteins that would initially result in the recipient, in the host immune system, recognizing them as foreign, so that they have survival and some degree of perseverance after they're administered to the patient, which allows them to hopefully and potentially function in their immune regulatory capacity in the way that you would desire. Next, please. And so in terms of clinical applicability, the target diseases are in general autoimmune and inflammatory diseases. And there has been a wide spectrum of clinical investigations for a range of inflammatory and autoimmune diseases. And if you hit next, please. Including clinical trials in things like graft versus host disease, something that happens after bone marrow transplantation, for example, Crohn's disease, multiple sclerosis, diabetes, type 1 diabetes, rheumatoid arthritis. Hit next, please. Heart diseases, if you think as an acute heart attack as an inflammatory event, and others. And there is now, if you hit next, please, approval in several countries around the world of the MSC-based therapy, particular for graft versus host disease. Again, a very devastating disease. It can happen after bone marrow transplantation, and it can be particularly difficult to treat in some cases, become refractory to other therapies. And this is where the MSCs are being utilized at present and approved for utilization. Next slide, please. So how does this work? So in this schematic, the MSCs would be removed from the body, all right, bone marrow or adipose, or in this case for NextCell, collected from umbilical cord donations. And then they are grown to large numbers in the lab. And this is very important from an industry standpoint because you need enough cells to be able to use in your potential therapies. And you'll see over on the left it says autologous and allogeneic. So autologous means coming from the same individual. Allogeneic means coming from a different individual. And the industry is best suited for allogeneic because you can obtain large numbers of cells from healthy volunteers. You can expand them in the laboratory and then have sufficient quantities to be able to utilize in your clinical investigation and hopefully your potential therapeutic. So the way it works then is that these MSCs, once isolated, once expanded in the laboratory, are then injected back usually into the bloodstream, and they will hopefully produce and release the factors that will target your specific disease. Now, if you look over in the lower left-hand corner, what happens to these cells once they're in the body? Do they stick around forever? Is there any long-term concern? And so what our current knowledge base is that the cells following intravenous administration will initially localize in the lung. This is the first major capillary bed that the cells will encounter, and the cells will stick in the lung. They don't become part of the lung. They don't turn into lung cells. And they generally will persist for approximately 1 to 3 days. While they are sitting there having blood flow exposure, they will sense circulating inflammatory mediators and be able to then respond accordingly in release of anti-inflammatory mediators. And then after several days, the host immune system does finally catch up and the cells are generally cleared out of the body. There have been a number of safety studies looking at long-term effects of MSC administration in patients for a variety of clinical indications, and there have been essentially no significant long-term sequelae to date that have been detected. Next slide, please. So in my world, I'm a professor of pulmonary and critical care medicine, and I'm strongly interested in the possibility of utilizing these cells in lung diseases and critical illnesses. So I'm going to show you some information from my clinical interest and my research interest that I think will be illustrative and then applicable to what NextCell is trying to do with type 1 diabetes. So in the published scientific literature, there are over 400 different publications in different preclinical models of lung diseases. And this includes mouse models, large animal models, explanted human lungs. And you can see in the schematic on the right that the spectrum of diseases ranges from acute lung injury to COPD, chronic obstructive pulmonary disease, or emphysema, bronchopulmonary dyspnea, a neonatal disease, asthma, and so on. And in every case in these preclinical models, administering the MSCs mitigates or ameliorates the specific disease endpoints that you produce in these models. Now, if you look at the list of mediators under each of the red boxes, each of the disease categories, these are different substances produced by the MSCs that have been implicated mechanistically in the actions, the protective actions of the MSCs. And you can see that there is some overlap, but there's some differences in the mediators identified for the different diseases. And this is a very important point, is that MSCs are not a catch-all panacea for everything you could potentially think to apply them to. They do have very specific functions and specific roles in specific diseases. Next slide, please. So when you think about lung diseases and critical illnesses, again, MSCs are not going to work for everything. It's very important that you pick your disease target very carefully. That's why I think type 1 diabetes is a strong candidate for MSC-based cell therapies. So if you look at the categorization of lung diseases and critical illnesses. There are acute inflammatory diseases, things like the acute respiratory distress syndrome, a very devastating critical illness that unfortunately has come much to the forefront with the COVID pandemic. This is what patients will die from, unfortunately. Sepsis, septic shock. These are acute high-intensity inflammatory diseases for which MSCs have a strong likelihood of having a beneficial effect. Then, next category is the chronic inflammatory disease. This is ongoing inflammation and or immune dysregulation. These would then include things like asthma and cystic fibrosis, bronchopulmonary dysplasia. And this is where the MSC administration has a strong likelihood of having efficacy. And type 1 diabetes would fit into this chronic inflammatory disease category. And then just to round it out, chronic fibrotic diseases or chronic destructive diseases, things like emphysema, things like idiopathic pulmonary fibrosis, permanent scarring of the lung, are less likely to respond to a cell-based therapy. Next slide, please. So in terms of clinical investigations of the cell-based therapies in lung diseases, most of the studies to date are phase 1, safety dose escalation. They're limited phase 2 efficacy. But you have to understand that, and this is very important for the consideration for type 1 diabetes, there still is no consensus on approach. There are MSCs available from bone marrow, cord blood, adipose tissue, and it's not clear if there's any particular advantage or disadvantage of the source of origin of any given cell. The question of autologous versus allogeneic, again, from an industry standpoint, allogeneic makes much more eminent sense. There are other considerations as listed, including dose and dosing, that we're still trying to best uncover. And this is why it's so important to do the type of clinical trial that's being put forward by NextCell. Next slide, please. And so just to give you a little bit more information, because I think this is important, the acute respiratory distress syndrome, respiratory failure requiring intensive care unit care, mechanical ventilation, there is no specific treatment, and there's still a 20 to 30% mortality. Now, in the schematic on the right, in the upper hand is a normal-looking lung. This is a schematic of the alveolus. And in the bottom is the inflamed, highly abnormal environment in the ARDS lung. Many different pro-inflammatory mediators, including cytokines and others, are part of the pathogenesis and eventual mortality of the ARDS. And so if you go to the next slide, we think that the MSCs have very strong effects in mitigating, and again, you can see in the schematic, that the MSC, once administered into the bloodstream by intravenous injection, will end up in the lung. It will sense all of those inflammatory stimuli and then release, as shown by the black arrows, a portfolio of anti-inflammatory mediators that will counter the inflammation and then result in clinical improvement. And again, this is supported by many, many, many preclinical studies, and we're beginning to see clinical investigations in ARDS that are showing benefit. Next slide. This is the same approach that is being applied in COVID ARDS, and there are currently now, to give you perspective, over 50 different trials, clinical investigations of MSC and other cell-based products for the SARS-CoV-induced acute lung injury. And we're all eagerly awaiting data from this. Next slide. Now, I'm going to shift gears a little bit to the science behind all this, because we still are trying to understand exactly how these MSCs work. And one of the important things to contemplate is that every patient is going to be different. And as Mona had indicated, in type 1 diabetes, the rate of destruction of the islet cells can vastly differ, or can significantly differ between different individuals. And so what we study in my laboratory and others is the effect of the in vivo, so the clinical environment, on the behavior of the MSCs. And this is a complex slide, and it's not, you're not meant to fully comprehend it, but the idea being is that we can collect biological fluids, blood or lung fluids or what have you, and then we expose the MSCs to those in the laboratory, and then we can characterize and determine what exactly the MSCs will then do in response to those different biologic fluids. What that does is give better understanding on how the MSCs are working in any given situation. And what that does is it helps do two things. First, it'll identify better a patient population more likely to potentially to respond to the cell-based therapies. Secondly, it gives you the potential opportunity to precondition the MSCs prior to administration to a given patient. And in some senses, this turns very much into a personalized medicine approach. It's a very practical approach, and one that we're hoping to elucidate for not just lung diseases, but for a range of diseases of other diseases, including type 1 diabetes. Next slide, please. How is this a practical consideration? Well, the title of the slide says it all. Cigarette smoke adversely affects MSC action. So in my world, in lung diseases, if I'm trying to treat someone who's actively smoking, it lessens the possibility of MSCs having a potential therapeutic effect. This is a broad-based, simplistic overview, but this is what we will hope to find out more about type 1 diabetes and which patients are going to be potentially better able to respond based on clinical and mechanistic behaviors related to their type 1 diabetes. Next slide, please. So to wrap this up in a way that I think is beneficial, the particular product from NextCell, if you look over on the right-hand side, is from umbilical cord. And again, this is a very potent source of MSCs. And again, there's no particular clear-cut advantage or disadvantage to umbilical cord versus MSCs from other sources. However, in terms of biologic activity, from an from an industry standpoint, umbilical cord is a desirable source because you have available source material, and what NextCell has done in terms of being able to manipulate these cells, in terms of expanding them and growing them in large batches, robust manufacturing, low variability, is something that Mathias and I have discussed, and is a strong platform for utilizing these cells. It also, if you look in the center, the cell therapy product, again, this is allogeneic. And so the data suggests that this is a successful approach, off the shelf, proven safety. In other diseases, there's a fairly strong track record of MSC administration in a wide variety of diseases, including in these very critically ill, sick COVID patients, and there have been no significant safety issues to date. And so easy administration, simply administered through an IV catheter. And so I think that this platform that NextCell is putting forth is a very strong one. I think type 1 diabetes is a good candidate for a cell-based therapy. And I think all the reason to move ahead with the next phase of clinical investigations that are being proposed. So Mathias, thank you, and I'm happy to answer questions as they come up further on in the presentation.
Thank you very much, Dan. Yes, so quickly wrapping it up. Next step for NextCell, our top priority is to bring ProTrans to the market in diabetes as fast as possible, and to then be able to investigate other indications to be able to expand the use for other type 1 diabetes than the ones that we have included in the clinical trials so far. So our focus is to design a trial with the aim to reach a marketing authorization approval. So we will consult the regulatory authorities and consult experts within the field, and also look at the FDA guidance and carefully design this trial, because we we want to prepare a robust trial with a significant number of patients. So we have been running very small trials, moving very fast so far. But at this time point, we need to carefully look at all the data we have generated so far, which is quite some data. The first patient was treated, it's soon 3 years since the first patient got the first infusion. So looking at the clinical trial program for diabetes, we have completed the dose escalation and the small randomized placebo-controlled trial. And our next planned trial is a randomized placebo-controlled trial in a much larger scale. We talk about in the range of 100 patients instead of the range of where we have been in 24 patients so far. Cell therapies are usually with quite fewer number of subjects as compared to other kinds of phase 3 trials. So when looking at other trials that has led to a marketing authorization approval within EMA, the phase 3 is typically between 1 and 200 patients. We also run the ProTrans repeat trial where we have given repeated infusions with ProTrans. So first we give one treatment, follow the patients for 1 year, and then after that we give a second infusion and follow another year. And all these patients have now been treated, and we will have been to their 12 months follow-up, and we are just waiting for the control patients to be at their 1 year follow-up. So hopefully we can produce or present the data from ProTrans repeat already this year. The main goal for this year is also to then submit an application for a phase 3 trial. But if we need more time to have the design, and that means that we have to wait to submit in early 2021, that is not a problem for us. It's more important that we really have the right design. And meanwhile, we are having discussions how to support investigator-initiated trials, treating other patients with the diabetes type 1 than the ones included in ProTrans 1 and 2. So by that, I would like to thank you and open for questions.
All right, thank you all for the presentations. We will conclude the event with a Q&A session during which we will address questions from the audience. So a reminder to send in your questions through the Q&A form, which is found at the bottom of the screen. But we will start by addressing the development of cell therapies with some questions to Dr. Weiss. So, Dr. Weiss, why are cell therapies, and particularly MSCs, such an attractive therapeutic approach to explore in comparison to typical pharmaceuticals?
So I think one way to think about this is that most disease processes are complex. And if you're fortunate enough to have a single pathway of inflammation or immune dysregulation that is amenable to a specific intervention, then that's a powerful way to go. But in most cases, that may only give you a partial therapeutic benefit or no therapeutic benefit because you have not hit the portfolio of dysregulated inflammatory and immune pathways. The strong and significant, almost unique advantage of cell-based therapy utilizing mesenchymal stromal cells is that they can target multiple inflammatory pathways simultaneously. And this reflects what they were evolved to do in terms of helping to maintain homeostasis. in terms of, again, inflammatory and immune regulatory pathways. And so the advantage of that is, I think, very strong. And I'll give you examples of trials, again, in my world of critical illness and utilizing that acute respiratory distress syndrome as an example. There is a strong platform of information in, for example, mouse models of lung injury in which single pathways, single inflammatory pathways, single immune pathways were targeted, and there was benefit in the mouse models. But then when applied to clinical trials in patients, no benefit at all. And it reflects, again, that mice are different than human, and that's a separate conversation, of course. But the underlying concept is the complexity of the disease process.
Okay. And so, in your experience, when it comes to clinical trial development process, how does that differ? Are there any characteristics separating it from the standard development of pharmaceuticals?
There shouldn't be, because you have to consider two major issues. The first is how regulatory agencies view cell-based therapy products as pharmaceuticals or not. The second is that any clinical investigation has to have the proper underlying statistical validation. And so this includes power analyses based on available data that would then justify and define the number of patients to be enrolled in any given investigation that will then give you the best chance of demonstrating a statistically significant difference between the intervention and the control. So from that standpoint, no, cell therapy trials are no different than trials from any other of any other pharmaceutic agent.
Okay, so an observation made is that the cell therapies trials are usually more limited when it comes to in sample size. Of course, that is also dependent on indication. But do you have any comment on that? If
well, I think yes, no, it's a good point, and Mathias made that point. I think that the what you just articulated is the most important determining factor. The disease indication for which MSCs have been studied include oftentimes rare or less common diseases, orphan diseases, if you will. Graft-versus-host disease, for example, is not common. It's devastating if one has it, but in terms of the prevalence, it doesn't compare at all to, for example, the prevalence for type 1 diabetes, which is affects many more people. So I think that the strategy that will best serve in the long run is to use an appropriately statistically defined large number of patients. Okay.
Yeah. And in your experience and with the NextCell's progress, what do you think will be important for them to demonstrate in upcoming trials?
Well, I would leave the specific endpoints to Mathias and Mona, as not being a diabetologist, but it would seem to me that any clinical investigation should provide mechanistic information as well as the hope of clinical benefit. And so the primary endpoints will remain safety, but also in terms of efficacy, the information that was presented earlier about levels of Cpeptide circulating in the blood, other diabetes, specific diabetes related endpoints are, of course, what you want to see. This is what the patient will want to experience. It would make sense to include quality of life type of endpoints, things like insulin use or episodes of hypo or hyperglycemia and the like. And again, I leave that to the diabetologist and the endocrinologist. But what I would also strongly encourage is that endpoints include measurement of inflammatory mediators in blood relevant to diabetes, other Cpeptide. And again, I don't know what those are, but again, what you want to best understand is in parallel with a potential therapeutic benefit is more information on exactly how these cells are doing what you hope they are doing. And with that information, you can then go back and refine the use of the MSCs, make it even more potentially potent for this particular patient population. And so I would hope that the endpoints would include, again, collection of blood, keep it quite straightforward, collection of blood at different intervals that could be integrated with the measures of clinical outcomes, collection of measurement of Cpeptide, for example, and then assess those blood samples for these other pieces of information that can only hope to make the product better in the long run.
Are there any parameters related to the cell therapy that you think will be important from a regulatory perspective?
No, I think this is a conversation that is best held with each regulatory agency in terms of how they define cell-based therapies and how they define the MSCs. In the United States, the FDA has a great deal of experience with this, and there are criteria that apply to cell-based therapy that are parallel from those from any other therapeutic. Straightforward things like sterility, reproducibility, reliability, and production capacity, et cetera. These all apply to cell-based therapies, and so there's no surprise there. In terms of specific issues with cell-based therapies themselves, one of the issues that's come up, and Mathias referred to this earlier, is that of potency. If these cells can make a portfolio of different mediators, you can speculate, you can postulate, but most importantly, you can investigate which of those is relevant and most critical for your particular indication. And so this is a type of information that as we hopefully learn more about what these cells are actually doing in type 1 diabetes, we can then go back and with the batches of umbilical cord samples that are obtained, select out even better the cells that are producing the things that you have identified as being most important in treating type 1 diabetes. All right, so the regulatory agencies would be keenly interested in that type of information.
Yeah. Okay, thank you, Dr. Weiss, for giving such good answers to our questions. And we now have some disease-specific questions to Dr. Landin-Olsson. So when it comes to, from a disease perspective, what is your view on ProTrans' potential as a disease-modifying therapy, and also in comparison to other late-stage therapies under development?
Yes, and there are lots of immune modulation that has been tried, and it is discussed that you should combine different agents, because when you strike on one pathway, perhaps the other pathway will take over, so you have a compensatory mechanism. So a good thing with stem cells is that you will strike on very different pathways, and you will influence a lot of ways where you can influence the immune system. And I think also it's very good that you have so few side effects, because with a conventional immune modulation you have quite serious side effects, and so this is a good thing. And when it comes to the evaluation, it is difficult, because you start on different stages of Cpeptide, and you have a remission phase where you can go up and down in your own insulin production. And there has been a lot of debate about this, and one thing is that you can measure how much insulin you need. If you need small doses of insulin, it's better than if you need high doses. But then you can have an insulin resistance in your body, so that will also influence these measurements. Then you can look at the HbA1c, So you look at how well can you control your diabetes, and you know that if you have own insulin production, you can control it in a better way. So that's one thing that you can follow. And when you come to the C-peptide, and that is difficult because you have different, and if you look at the percentage, how much you have lost, it also dependent. If you have very little from the beginning, it will be the percentage will be high, and it will be very sensitive to what you will lose. Yes, so I think you should go further with a big trial, and you should have a lots of endpoints which you can measure. C-peptide is one, but you should also have a lot of other things to measure.
Are there any other certain aspects of such a trial that you think are important to address when it comes to design and setup?
Yes, I think it's very good that you have humans, because there are so many trials and studies done on experimental animals, especially baby rats, which is an experimental rat model for type 1 diabetes. And I think that is that rat is very different from human, and most things will work on that rat, but when you come to humans, it will not work. So I think it's right to go to humans and do human studies. And as Professor Weiss pointed out, it's very important to have a mechanistic to study the mechanism, how it will work and how it will influence the immune system, that you measure that and see if you have markers for immune system and the immune activity.
Mm. So and in order for ProTrans to fit in clinically in the commercial setting and for patients, are there certain characteristics that you think are important for the product to have?
In the study, or?
No, when it comes, if it reaches the market. So in order for it to fit into the clinical flow in the therapeutic regimen, are there any certain factors that are important in order for it to be viable as a therapy when treating patients?
Yes, I think on an ethical view, I think it's important to start with adults. When you come to adults, you have the progression of a destruction is much slower. So you have time to study what is happening, and you do also have remaining insulin production at the start. Because if you go to children, the process is much more rapid, and the chance to interfere, it's much less.
Okay, thank you, Dr. Landin-Olsson. We finally have some, or lastly have some questions to Mathias. What are your main conclusions for proceeding into later stages clinical trials?
Yes, we have very consistent data from the two small trials that we already conducted. And we, of course, have a lot of secondary endpoints that I have not gone into, which are the ones that Mona raised with HbA1c, and we have insulin requirements and a lot of things. The golden standard is C-peptide area under the curve, and that was our primary endpoint, and we have so far only presented the primary endpoint. It was small sample size from the beginning, and even though that we have individual differences, we see a very clear difference, statistically significant difference in this small population. So we need to go continue with the larger trials to have these individual differences, to have them how to say, in order to make when having a lot more patients, we could detect the difference much better and with higher statistical significance. And when looking at the trial size, if I may share a slide again, the trial size these are totally different cell therapies for different indications, but it's just an example of the sizes of the trials that have reached EMA approval, and we have three different here that have between the top one has 75 in open label 3 doses, but then the phase 3 is only 102 patients. One of the last approved drugs, Alofisel from Tigenix, had an open label dose escalation with 24 patients, which is equivalent to what we had, but we had the phase 1/2 as a Simon 2-step trial. So we are having relevant number of patients for the cell therapy clinical development. And the phase 3 would probably require somewhere around 100 patients. So the next logical step is to go through with a phase 3 trial or a pivotal trial. And we will discuss this with the authorities.
And so cell therapies are typically limited in scale based on, well, for example, production cost and pricing. So what is your approach to this, and how will ProTrans be commercially positioned?
So we have now a production that is relevant for clinical trials in a larger number. So and we also investigate the possibility to use bioreactors instead of two-dimensional cell culturing. We recently got a Eurostar grant together with a Dutch, a Swiss, and Belgian company to evaluate the bioreactor production of ProTrans, which would increase the scale and also the operator, make the operator less variable, so we could optimize this production. Cell therapy is not as producing small molecules, so it requires more material and it has more limitations in the scalability, but the design of our drug product is taking this into account. We have used, while designing our product, we have thought of the scaling possibilities, and we use a source that generates a very large number of cells per donor. We have a source that we can easily obtain. The willingness to donate umbilical cord tissue is very high. And we produce these batches with pooled cells. So we use cells from multiple donors to generate larger batches. So in terms of scalability, we are very well positioned, and we are further investigating bioreactors for this.
Okay. Would you like to conclude with the event with some final remarks or take-home messages?
I put this slide here within similar trials within type 1 diabetes that I thought was very interesting. Here is the different antibody trials that has been conducted during the last 10 years. And we see that by using antibodies to knock out specific immune cells, they have achieved to keep a lot of the Cpeptide, but it's not that impressive. The treatment benefit is somewhere between 20 and 50%. However, these treatments are rather harsh. the CD3 treatment, all the patients has to be hospitalized for 14 days, given a lot of infusions, and they have a lot of severe adverse events. Our treatment is a single infusion. The patients are discharged from the clinic within 3 hours. And in our small data set, we have achieved 80% effect, or remaining 80% more Cpeptide than the placebo-controlled group. So I'm very confident moving forward in a larger pivotal trial.
Okay, well, thank you everyone for participating, and thank you to the audience for listening in. We will conclude the event now.
Thank you all.